Journal Club, Department of Biotechnology, St.Marys College, Thrissur. Presentation is prepared by Group 2 and the members are Ashwati J, Devika Ashok, Divya VR, Lakshmi Ajit, Sangeeta P from 1st BSc Biotechnology 2019-2022 batch. We are here to present the presentation on the topic Qualitative and Quantitative Analysis of Drinking Water Samples of Different Localities in Abbottabad District, Pakistan. As we all know, water is the most widely distributed and abundant substance found in nature. The quality of water is a vital concern for mankind, and at present, waterborne diseases and epidemics still booms large. And coming to our topic, we have taken 15 drinking water samples consumed in different areas of Abbottabad district and analyzed it completely. Both the qualitative and quantitative analysis of water samples of different localities was conducted to determine the exact amount of different pollutant present in water. These water samples were taken from the main water sources where maximum people were using them for drinking purpose. And the result of the research work showed that the collected drinking water from different areas was not suitable for human health due to microbiological issues.
Materials and methods. Materials. Chemicals involved in all chemical tests were analytical grades such as HPLC grade solvents. Collection of samples. The selection of sampling point is more important than the actual chemical analysis of water for the quality investigation purposes. Here, the samples are collected in a polystyrene bottle of 1.5 liter capacity, which were washed thoroughly with detergent, acid, tap water, and distilled water. Measurement of pH and temperature. pH is measured by using portable battery operated pH meter model MP220. pH of the sample should lie between 4.0 and 7.0. The sample temperature is determined at the same time. Alkalinity. Alkalinity is the measure of hydroxide and carbonate ion content of water sample by titrating it with standard H2SO4 using indicator. Endpoint pink to colorless. Total dissolved solids. Here solids refer to the matter suspended or dissolved in wastewater. The sample is then measured using a pre-calibrated conductivity meter model in OLAB count level 1. Electrical conductivity. The conductivity of sample was measured using a pre-calibrated conductivity meter model in OLAB count level 1. The measurements are taken at room temperature. Total hardness. 1 ml of hardness buffer solution was added to a 50 ml of water sample, followed by addition of 1 to 2 drops of indicator. Then the solution is titrated against a EDTA solution from a burette. Endpoint reddish to blue color. Therefore, it can be calculated by a formula that is total hardness is equal to worsenate solution into Mi of the sample.
Determination of chlorides. Presence of chloride ions is tested by using a solution of potassium chromate as indicator. Here a brick red precipitate of AgCl is formed. It indicates the endpoint. It is because the silver ion reacts with chloride ion, forming brick red precipitate of AgCl. Then moving on to turbidity determination. Turbidimeter or nephelometer was used in this experiment to determine the turbidity. Here formazine primary standard was employed to calibrate the nephelometer. Then total coliform count. The typical coliform colony has a pink to dark red color with a metallic surface sheen. The sheen area may vary in size from a small pinhead to complete coverage of the colony surface. Typical colonies can be dark red or nucleated without sheen. Calculation of the coliform density is as follows. That is the total coliform colonies per 100 ml equals coliform colonies counted per sample used in ml into 100.
Results and discussion. The result is divided on the basis of three parameters: physical parameters, chemical parameters, and biological parameters. The following are the points that comes under physical parameters: temperature, pH, total dissolved solids, conductivity. Temperature. Temperature of water samples was taken at the spot in February 2010. Temperature range of sample from different places varies from 30 degrees Celsius to 17 degrees Celsius. Average temperature of water of Abbottabad district is 14.9 degrees Celsius. The maximum permitted standard of drinking water is 25 degrees Celsius. So, the water of Abbottabad district is suitable for drinking purpose. pH. pH shown by water samples taken from different places of Abbottabad district ranges from 7.26 to 8.84, which seems to be alkaline. The standard of water quality on pH lies in range 6.5 to 8.5. So, this study shows some water samples are exceed this limit set by WHO. TDS, total dissolved solids. Maximum TDS found in Abbottabad district well water is 664 mg per liter and minimum TDS is 198 mg per liter. As the TDS range set by WHO for drinking purpose is 500 mg per liter, the results show that water of Abbottabad district is fit for drinking in terms of TDS. Conductivity. In many cases, conductivity is linked directly to total dissolved solids. Conductivity of drinking water by WHO is 5 to 50 millisecond per meter or equal to 50 to 500 microsiemens per centimeter. Mean conductivity of drinking water sample of well water is 683.24 microsiemens per centimeter. Maximum value is 132.7 microsiemens per centimeter. Minimum value is 395 micrometer per second. The next one is chemical parameters. Under chemical parameters, we have calcium hardness, turbidity, chlorides, and alkalinity. Calcium hardness. Calcium hardness of water samples collected from Abbottabad wells. Mean value is 298.13 mg per liter. Minimum value 0.160 mg per liter. Maximum value 0.350 mg per liter. Calcium hardness range set by WHO has a permissible limit 200 mg per liter and maximum limit 600 mg per liter. So, obtained results comparing standard value of calcium hardness are found to be less than permissible limits set by WHO. Turbidity. Turbidity range set by WHO 5 NTU to 10 NTU. Turbidity range found in Abbottabad district water samples. Minimum range is 1.74 NTU and maximum range is 20.02 NTU. Chlorides. Chloride found in well water is 10.44 gram per liter. Chloride found in water sample is found to be 0.8 gram per liter. Maximum range of chloride is 26 mg per liter. As the chloride limits by WHO ranges from 250 mg per liter to 1000 mg per liter, no samples cross this range by WHO. The water of Abbottabad district is considered suitable for domestic purposes. Alkalinity. Alkalinity range by WHO is 500 mg per liter. From the water samples of Abbottabad district, alkalinity found in well water sample is 300.4 mg per liter. Minimum alkalinity is found to be 120 mg per liter and maximum alkalinity is 476 mg per liter. Therefore, it is found that the water from Abbottabad district is suitable for drinking purposes. The next one is biological parameters. Total coliform count. Total coliform range set by WHO is 100 milliliter raised to -1. Minimum coliform count found in Abbottabad district water sample is 15 per 100 ml and maximum range is unlimited.
As we know, the basis of this experiment was to find out whether the water samples collected from Abbottabad district of Pakistan were suitable for drinking or not. Going through the results, we have come to the conclusion that this water is not suitable as it is extremely harmful for human health due to the microbiological issues. There are some suggestions or recommendations to cope with the problem of this crisis. Number 1, water can be boiled before use in domestic purposes. Number 2, X-rays can be used as a water filter for clarifying the water from microorganisms. Number 3, if the contamination level is extremely high, we can perform chlorination only by an expert chemist because excessive chlorination is harmful for human health. Number 4, efficient system for garbage collection as its disposal outside the city areas. And number 5, sewage waste treatment, amongst other things.
Journal Club, Department of Biotechnology, St. Mary's College, Thrissur. Presentation is prepared